Sarkar, our Dean MBA Professor Dr. Makaut sir, and all my faculties and dignitaries. I, Dr. Ghosh, now today working as moderator of this session. We feel proud and privileged to get Professor Dr. Jaya Bandyopadhyay with us as the research person for this session. Professor Dr. Jaya Bandyopadhyay serves as professor in the Department of Biotechnology and Director of the School of Biological Science and Technology at Maulana Abul Kalam Azad University of Technology, West Bengal, Makaut. With more than 23 years of teaching experience at Makaut, her expertise spans precision medicine in cancer, cell biology, molecular development, and nanoscience. She earned her PhD in geology from Vishwabharati University in 1995 and spent 6 years in South Korea completing postdoctoral training at Chonnam National University, Gyeongju Institute of Science and Technology, and Hanyang University, Seoul. She also worked as a CSIR Senior Research Associate at the Indian Institute of Chemical Biology, Kolkata for 13 months. Professor Bandyopadhyay has supervised 7 PhD scholars. Among them, 5 of them has already received their degrees and 2 PhD degrees are ongoing. Conducting research on molecular aging, nanoscience, stress-induced neurodegeneration using C. elegans and mammalian models, and precision medicine in cancer, in vitro and in silico approaches. She has published over 50 research articles and authored 7 book chapters and also led several R&D projects. Her accomplishments include the Shiksha Ratna Award 2022 for her distinguished service for the cause of education conferred by the Government of West Bengal, and the Brain Pool Award funded by the Korean Federation of Science and Technology Societies to work as a visiting scholar at Gyeongju Institute of Science and Technology, South Korea, and Hanyang University, Seoul during 2007 and 2008. Now I request coordinators of this FDB, Professor Dr. Sarkar, to come on the dais, and along with co-coordinators, Madam Professor Musmi Atho and Professor Uttara Basu, Madam, to please come on the stage. Now I request Dr. Makaut sir, Dean MBA, and Mr. Indranath Mitro, AGM CRTT of SKGI, to join the felicitation program of Professor Dr. Jaya Bandyopadhyay. Please, Madam. today's topic is enhancing the efficiency and precision in the field of pharmacy through AI. Now, artificial intelligence is bringing major changes to the field of pharmacy by improving speed, accuracy, and patient safety. This talk explores how AI is being used in areas like drug discovery, prescription handling, medication adherence, and pharmacovigilance. Real-life examples and simple explanations will help participants from both science and engineering backgrounds understand the role of AI in pharmacy. Special focus will be given to how engineers contribute through AI models, smart devices, and automation. This session will also discuss ethical concerns and the importance of human oversight. By the end, participants will gain a broad understanding of how AI is shaping modern pharmacy and the opportunities it offers across disciplines. So, please, madam.
Thank you. So, a very good afternoon to all.
Good afternoon.
And I hope I can keep you awake after this heavy lunch session. I myself have to also stay awake. Also, I had a very, very heavy lunch because according to me, it's quite heavy. It was heavy. But anyway, it was really a great pleasure. And I'm really happy that Devashish extended this invite to me. And I was, in the beginning, I was slightly skeptical. How am I going to manage this session? Artificial intelligence in healthcare was the topic, right? And then when I got to know, I accepted it. I accepted it in the, I think, in the very first day. But then I was not knowing what I'm going to do because for different individuals because of this. So research is still going on, but I think that it is being in America now they are practicing. For that, the depth of research is going on. The same drug having two different effects is being established, and it is in every journal. One or two articles are coming out. But yes, of course, at the laboratory level, it is being proved. It will definitely come out in the market. But I will not be able to really say anything beyond this because it's not that because I am not a person who is a pharmacist from a doctor's point of view. You understand? But we have seen that the same drug, why it is having different effect because as I told you in the beginning, a drug is having a target. It should go and bind. Okay. Now this target for you is something else. Another person, it could be something else. This who will decide? Research will only by some laboratory test you will be able to find out that yes, maybe the genetic background is different, slightly different from one to individual. That can be an effect. So I will come into it. I'll come into it. So more in details, like personalized medicine, when you are trying to prescribe a drug, not generally. You can now prescribe a drug individually. It's absolutely tailor-made for a person. It is possible, and that is what the research.
Outside India, there are countries who practice means doctors say that I need these, and pharmacists are the best person to say that if you want to cure this patient, this can be a best composition. So that's how different countries practice
this is what we need
But
to communicate.
India goes there is like paracetamol,
This is
600,
very
200 and all.
This is important.
That's a very bad.
The communication between the doctor and the pharmacist and and other people, they have to be there just for making a drug to be prescribed for a particular disease. Absolutely, you are correct. But then, see, we need to also change our mindset. So this is what I actually I will this personalized medicine that I told you, that one size fits all approach is now slowly we are going to deviate from it. And this I'm going to talk a little bit later. So that's why I kept the slide in the beginning. And I want to quickly move on to some other advantages of AI. I think these are some of the very common slides which I think you will be listening and looking into in the next few days lectures. All of these will be often mentioned. So clinical trials And any monitoring, as ma'am said, that real-time monitoring and all, this is definitely the attributes of AI. And of course, the outcome is obviously good. So that is why AI is now becoming a part and parcel of our life. AI has really made a hospital taking decision very quickly on the outbreak of flu in Orissa. So it was like, this was in news, and also there was another one. There is another one. Maybe, okay, I'll come into it. So having AI in your hospital where it is going to, where the flu was detected, and they were able to manage that crisis because of AI. Now, automation and all, I think all of you will agree that AI has immense attributes with regard to all these which are done manually in the different companies. So I think I can skip these because these are some of those common and general views about AI in pharmacy. Now the supply chain. Think of the supply chain from the manufacturer, from the grassroots level manufacturing and coming to the patient. There is a way of handling this. Like somebody is giving out the raw products, it's going to the factory, they are formulating the medicine, and then it is going to the pharmacy or with the company, and then they are making it into a drug, formulating in the drug, and then finally selling it off at the patient. So the entire process is definitely there, and it is already there. But if AI is taking charge of the entire thing, think the amount of the pace with which this supply chain can improve. That I think I am no one to predict because I cannot say it in values, but you can imagine how fast it can be. So obviously we should definitely be very appreciative and be positive about AI's role in pharmacy, especially in precision and efficiency. So even Pfizer's drug, which could have taken like 3-4 years, within 1.5 year, they could create the drugs or manufacture the drugs and supply it to the patient population. So productivity has increased, and also the cost has reduced for all this, and the time also required also is like kind of means lesser time, and production is getting improved. Let's move. So this is another same. I can move on. I want to now one thing. Now the engineering. So this talk is going to be like pharmacy driven and delivered by a biologist. But of course it is for you all engineers, my respected engineers. So where is your role? You have to now, I will give you some tasks later on, but wait for a while, that all the computer science engineers, all the electronics, because I think many of you from the electronics background, computer science, data science, mechanics, I think all of you have immense role in developing this technology. What you require is what we want. You have to gather information from the doctors, you have to gather the information from the biologists, and then you integrate all those information and try to see whether you can make out a package, a kind of a package, and bring it out into the market. So we cannot direct you how the computer science circuitry will be required. You will know. Means the electronic circuitry will be required. You know the programming. You know all those, but you need the real-time data from us. So there is this interdisciplinary approach. You cannot survive if AI has to be there. It has to cater to the human mass. AI will not be there for only an inanimate object. It is going to cater to the human mass. And human mass, there are many other applications. But when I am talking about health, then it is a health, your body, your cells, your tissue, everything. So you also have to understand, all engineers have to understand the language translate whatever the cell has to speak and you try to understand the cell, then you will be able to master this technology. So here is the role of all the engineers. So I am saying that this is an interdisciplinary approach and collaboration. So just kind of remember this for a while, keep it in your mind. Why? Because at the end I have something to offer so that all participants from different background, I do not know whether it will be possible or not, but if you had more health care or pharmacy background participants, then we could have done efficiently, but let's see. Let me work it out. Now, here is something that I want to not quickly because I want to move into my topic very fast. But these are some of the languages that are practiced in understanding AI. And this LLM or this GPT, generative pre-trained transformer, I'm being very honest with you. Preparing this lecture, I learned many things. Obviously, ChatGPT everybody knows. I even didn't know that ChatGPT generative pre-trained transformer, this term, it was there. I had to find out what does it mean. And I found out, okay, GPT means this. So, see, preparing a lecture also has trained me in a way so that I can communicate with you. That's a good thing about having all these short lectures and seminars. So now these, and I did not know how it was operating. So it requires some language platform, which the engineers will be able to explain better than me. So these language models, whatever is there, for pharmacy, how are you going to implement it? You are going to either speak in front of a machine and speak about your disease, or say I am having this problem, and there will be a response from the other end, which is an AI. It's all AI driven. So sometimes it is correct, sometimes it is incorrect. You have to be very careful what to take and what not to, because you might then you say, ma'am, then what is the use of AI if there is incorrect? No, because an incorrect information or output that is coming out because of you. You have fed it all wrong information in the past. So do you understand that if we are making mistakes, response will be accordingly likewise. So these are some of the bottlenecks we have, and we have to be very, very careful on feeding whatever the information, because as I told you in the very beginning, the brain analogy, whatever we are trying to understand by the pattern, if we are feeding all those information in the correct way, the output will be correct. So we have to be very careful about all these information. So ChatGPT is now in all pharmacy. I think ChatGPT is not in pharmacy, everywhere. Everywhere. Without which I think many of you might not be able to even survive. It will seem that your life has become simpler. Why not? Because now I want to tell you one thing. You might think that no, ChatGPT is really ruining the kids. I am not talking about the kids. I am talking about in the positive way. If you can reduce your work and you can concentrate on some other job, like AI, what has happened to all the pharmacists and all the doctors, they are now, because all this drug and every implementation has become so, they have reduced the time, they can counsel. They are spending more time in counseling. That is the result of having AI in the medical field, because doctors, pharmacists, they can concentrate in counseling, which was not there. Human touch is required. I'm not saying that AI is the only thing. Human touch is there, but there should be a balance. There should be a what to take and what not to take. It is up to you to decide. So therefore those ethical issues are there. You must not be biased in your data. You should always feed in all unbiased data. Means you should not make any mistakes, because that is going to ruin your output. So all these errors, privacy concerns, patient data, patient information has to be kept in secret. You cannot, in confidential. You have to leave it confidential. So these things have to be there, because I tell you, we access human data from the servers, and we generate and we carry out our research by taking the data from fully concerned with the incidence of that disease or whatever the mutation has caused the disease. We are only interested in that, and then we will plan our research accordingly. So these things have to be really taken care, and AI, these are something that all of you should know. So human oversight means what I have missed out. I made a mistake. I did not see it, and I made a mistake. So those cannot be actually ignored. Human oversight is always a machine oversight is slightly less. Human oversight is still there. Machine might not do it. So we should always mitigate all these problems by. Oops. Okay, let's. So actually oh yeah, let's come back. We wanted a break because now we stop. Just about 1 hour. I'll wrap it up. Don't worry. So let me go on. I can continue because just listen to me. So you should all be very, very particular about the data that you enter, whichever module. So it is up to the students, it is up to the faculties, and also the experts in the field who are dealing with the artificial intelligence and the different modules that are there. The different types of AI are there. You all have to be very careful with the original data. This machine learning, what do you mean by machine learning? Machine is going to learn from the data that you are in. I will have a, what do you call it, this ECG report or any tissue. Just, I give you an example. Just think of it. Have you ever thought about it? I have seen, I have tested it. A medical report or any research tissue. You know, you have all these slices, tissue slices, and then you do some histochemistry, and you take those tissue and doctors have to prepare a report out of it. You know that. What they do, AI has made it simpler. They are sharing those data across the different countries, and they are trying to generate the report, taking help of all these different units set across the globe. You take such tissue, and you can also generate a report because you just put it into the ChatGPT. I have tested it. I have seen it, and it is going to give you the report that any histochemist would do it. Whatever is the defect in, say, lung slices or heart tissue, you will get the report. That's because how? Machine learning. Whatever the ChatGPT or any such chatbots, I don't know if there's any other there because I am really not an expert. You people can tell me. If you just show they have already learned that is a tissue of a heart and not lungs, and it will give you the exact report. Sometimes it can make mistakes, but I have seen at least 2 examples I saw long, like 6 months back, I found it was correct. So you understand how machine learning can make your life easier. Means for the doctors also, you can get it, but I'm not saying that you have to do it. I'm just giving you the examples of such usage or implementation of AI. So let me quickly move on. So all these are all, I think I have covered most of it. They are all very common, general platform. What are the approaches? See, always remember that whenever you have a medicine, you must test the efficacy of the drug. You need to test the efficacy of it. That is important. And if the drug efficacy is being proven by some platform, before you treat, you know that the drug will have technology to the society. So if it is for healthcare, you should start. Then only it can, and then start teaching or give training to the other mass. Gradually build up your team and start the training and education, and then gradually it can be implemented. But do not forget that research and development R&D is also required, because it's about health. So one experiment or one data cannot be enough. You need hundreds of data to be able to come to a conclusion that yes, this is the outcome or this is the drug is going to have effect. And finally, in this career pathway, you can be a leader also. So this is what I think can be the roadmap for all the entrepreneurs. And so I can skip this, because this is again, you have degrees, different degrees, because this is a pharmacy, because it's in with pharmacy, so obviously your bachelor's, master's, doctor, PhDs in pharmacy, that degree is definitely very much important, because without it you will not be promoted to the next level. So those are the basic requirements, and you need to have the skills, all these skills of pharma knowledge. Then okay, Python and R, those are important. Now I do not know Python, but my sons, they know. The point is that I did not require it, but if required, if I might have to study at this old age. But anyway, this is required. So you see, nothing is redundant, or nothing is that non-essential. We are all required. Each and every person sitting over here can contribute to the best for the mankind, and in a proper way, with all the knowledge and expertise that you have. And machine learning, data analysis, I think everyone has that. You have all papers. You have all papers in it. Now, and this interdisciplinary communication, which I am going to talk about. So, drug discovery, digital pharmacy. I think many of you know digital pharmacy. Now you can get your medicine sitting at home. And even Government of India has also made, I have a slide on it, that Government of India has made some platforms where you can easily order. Okay, so therefore our health care at the bedside is very, 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 very close by, and we are gifted that we have such people around us who are appreciating these type of endeavors from the government and as well as from ourselves. So India has this telemedicine. I think there is a unit. There are people who are recruited for telemedicine, for promoting telemedicine. I have not done it, but I really want to do it. I heard that it is really working very, very effectively. So other countries are doing it, but definitely we should know about our own country, so it is there. Next is, these are some of those Arogya Setu. Then you have e-Sanjeevani, all Government of India initiatives for reaching out to the patient, be it rural or be it at the urban area. So we have to know how AI can really reach out to the common mass, because that is something.
There is a lot of things like telemonitoring and all those things.
So you are from healthcare?
Yeah.
So she is the one who can really understand what my language over here. But anyway, you can understand what I wanted to share. So therefore, and these and somebody was saying that the regulatory bodies, FDA approved, they have to be there. Sometimes they have this out. So you need to adhere by all the international standards and electrochemical standards and this Food and Drug Administration. But data protection, that has to be also there. So you have to sign, or before you even come out, you need to sign all these, because data protection is something that you have to really be very, very careful. So if we are abiding by all the professional guidelines, all these regulatory guidelines, why not just go ahead and venture into the AI? So, but I want to say that, okay, AI is there, a machine is there to take care of me. Does that mean that AI is going to replace human? No. AI will not replace human. Human will be there, job. No. You must find out what is your strength and try to see where you can contribute. So these are some of the limitations. You all can understand what are the limitations. This is nothing new. Now I want to really move on into the most important topic. So reaching out to the urban hospital, that is telemedicine. These are some of the advantages or the applications of AI. And bridging the digital divide, since it is AI, it is all information through machine. Obviously, you have to reach out to the common people. So if they do not have any access to such technologies, then this digital divide is there. So that is one problem, and we have to see how we can solve that. So connectivity issues, and I think you all understand during the COVID pandemic, even giving out a laptop or a small this, what do you call it, this tab, you're giving out tab to the students was a big problem because it requires money, but there was this digital divide between the rural and the urban, so you have to think how you can solve that. So I will now move on to the future is now why we must prepare. So this is something we are prepared now, that something is going to happen in the next 1500 years. Something remarkable is going to happen. Right now only it is happening, and you can predict what is going to happen. So just think of it that we have already started all these prescription verification now already has started. Personalized medicine still, as you say, that it is now going research, but yes, it's going to take place. But so we can expect that the next 5-10 years, we can expect that individual medicines can come out, and we will see all these wearables, advanced wearables during this period. And this 2030 and beyond, you can think something remarkable. I really do not want to even think, because I feel that I will be an animal in the zoo cage, and some super AI is going to come and watch me, and I will entertain that super AI. I really do not want to really think, but it is something that we would like to look forward to. So there are many limitations. So and now I want to, these are something just with the flow it was there. And funding, of course, you have to remember that all about this funding. Who is going to fund? Without money, nothing can be done, especially as I told you, that the initial investment is always high. The down the line, it will be always very, very effective to the commoner, but initial investment is high. So you need some projects, own projects, handsome projects, where you can really engage some students to work on a problem which you have devised, or it is always you not alone. You should always make it an interdisciplinary. Without taking help of other experts, I think a good project will not come out successfully. Sitting alone in one corner of the country and doing research without communicating with others has no value until and unless you have an interdisciplinary approach. So India has already given a lot of, I think all of you know which are the government agencies that you can go forward and apply for projects. So go do it. Now this is a part which you can do it, everybody will come. Now I was really not interested to teach this. Now is the part which I want to really focus. So if you have any question?
World Bank is providing this kind of.
Yes, I tell you, World Bank, TechIP for our government agencies, TechIP is there, we also had. So I might have missed out a few, but these are some of them which came into my mind. I have listed them out. So please approach to all these funding agencies for some novel ideas, innovative ideas. You are bound to get a project, I'm telling you. We are, what they say, the funding agencies, lack of ideas. Lack of ideas. Now lack of ideas means what? You're not going to copy somebody's idea and put it as if it is yours. No. Try to find out something something which is going to really help the mankind. Something absolutely apply to the mankind. Don't our mind. It's not possible. Although we are intelligent, but we're not that intelligent also. So I have something now. Next, I want to go into this personalized medicine. Now, next 15-20 minutes, I'm going to bore you with my some research, but I think it will be interesting because this is something that I wanted to share, and because it's a faculty development program, you faculties have to take something back, something which will be thought-provoking. All this what I showed you is everywhere you'll find. Now, I have done nothing to add anything extra. Now something I might add a little bit extra, or might be interesting, or you might know or may not. Let's see. So this was the idea. This is that slide I wanted to say, from one size fits all to precision care. You know, I just want to tell you, my scholar Shohini, she's an MTech student, and we have MTech programs. So she wanted to carry out her PhD under me, and she said, "Ma'am, I want to work in cancer and precision medicine." I said, "Shohini, I am not a cancer biologist. Neither do I know anything about precision medicine. I cannot be your guide." "No, ma'am, you just allow me. I will share my ideas with you, and you just give me the platform, because your expertise in different fields I've seen. Now, you know, as biologists, we are always, you know, you are working in cancer, you will work on cancer." And now I said, okay, I work with cells, but I do not really work on cancer cells. It's oncologist. I won't say I am an oncologist in the medical term. She gave me that assurance that she will work it out herself. And then she said, "Ma'am, I have a long time of precision medicine." I said, "Personalized medicine. You know this term, precision medicine, personalized medicine. All these jargons that we have, they're very fancy. Nice to hear." I said, "Oh God, now you're making my life more tough, because my work has nothing to do with personalized medicine." And she said, "Ma'am, you have expertise in cell biology, you have expertise in molecular biology. Why not give me the chance? I will share my ideas with you." So that's how my entry into precision oncology came. Now you can understand by the term precision that means something very specific. So what was her idea? She wanted to work with some phytochemicals, plant extracts. You know, this is always everywhere. If you're from pharma background, you would have gone through many papers with a lot of plant products, plant extracts working as medicine. We also take plant extracts for our medicinal purpose. So we cannot actually ignore the fact that plant extracts are just, means they have no use. No, only chemical drugs are only useful. That I do not agree, because many people, they still depend on Ayurveda. Okay, so I think this is up to personal choice. Who is going to select what? So let us leave the choices with the people. Let us see what we can offer. So what was her idea was that, so as I told you, precision oncology, the same drug might not affect the other individual for the same disease. So there will be something different in that individual. So we can understand that there are many, as I told you, DNA at the DNA level, or some other protein level. Somebody's responses are different, gene expression is different. So all these factors, this is just to make you understand that if there is from the patient, finally you are going into the targeted therapy, which all the data that you obtain from the patient, then you analyze it through your digitized, everything is digitized. So you analyze whatever is there from the patient, then you have an analysis, and then you bring it out into a server, or the data has been posted. Now it is researchers like us, we have to capture those data and try and sit back and see, okay, so this disease has this mutation, and the same disease, another, they have different mutation. How is it possible? We were thinking that mutation should be all same disease, same mutation. No, that's not true. So now when Shohini came in, and that time I had some MTech students, and it was really tough for me because I had to move on to a new topic, which was very challenging. See, we want to be in our comfort zone. Whatever we have been working for the last 20 years, we will still work. Taking challenges is something that human beings always try to avoid. Then I said, okay, then make a paper, do a review. So that review, they did it long time, it was taking, so finally they got it published, just recently, last year maybe, 2 years back. So glioblastoma is also another kind of a brain disease, and where the cells start to like degenerate, and you know you will have a lot of like symptoms. You can have speech problem, you can have memory loss, and all that. Brain is a control, so you can imagine what sort of problems can arise. Yeah, glioblastoma is the glial cells and then the tumor. Now the tumor spreads, and you know finally you have all these symptoms. Eventually, it's a really high degree cancer, and patients die. I had one friend who means a friend, I would say, a kind of a colleague type. He died of GBL. The fourth grade, it's a higher grade. You know, the grades are already defined by the doctors. So this is Shohini's project is glioblastoma. Now I said, okay, my lab has been already working with neuronal cells. So I said, okay, I can allow you something to do with the neurons, the neuronal system. So here was now I want to tell you. This might be very absurd or vague to you, but this is what we see inside a tumor cell. And you know, all these points that you see, we call them players. You know, these are all proteins. Players means how? Football ground, you have players to play. So in the cell, these are the players. The proteins are playing their role. So out of that, you know, there are many proteins. And finally, the proteins will behave in such a way, they might have their normal physiology, or they might go for a cancerous or a disease line. So this is, I think, in biology, everybody understands this language. So Shohini wanted that, I told her, see, I can give you on one condition. Don't study something that is very general. Go for a target specific. Now what is a target? A protein has to be there, which could be a marker for cancer detection. Now, and she started to study. Obviously, she is a research scholar, joining first time with me, and in cancer. So she found out that in glioblastoma, there are many proteins that levels go higher, and if they go higher, cancer is like detected, and it is like kind of a marker. We call it biomarker. Means in medical jargon, it is called a biomarker. But you know, saying a biomarker and publishing it is very difficult, because you have to be really sure that the biomarker is real, if the levels go higher or if the levels go relatively lower, and it is an indication of the disease. So she found out that there was one gene, the protein, which is called EIF4A3. So you can see one EIF4A3, this gene. She found out that this gene expression is very high in some glioblastoma. And then she did a little bit of studies, and she found out that not much has been worked upon it. She just told me, ma'am, I found out that there are many other genes, PAP kinase, there are many proteins, but this gene I found out is found in the glioblastoma, and I cannot find out any other much reports. Then she said, okay ma'am, this is going to be my target. Because as I told you, that be very target specific. So target means a protein. So that was kind of a story. I cut a long story short. I'm not going to go into the details of what was her plan, but she could find out that this EIF4A3 is a protein that has a positive role in the normal physiology of the cell, but if its expression goes high, it can cause cancer. But that has to be proven with some experiments, and there are some reports, but she also has to do it in our lab. And the next question is whether I can go for some plant extracts which can actually minimize or reverse the cancer-like phenomenon. So that was how she entered with this project in my lab, and the first thing that she had to do was the computational analysis, that in silico which I told you, which requires a lot of AI and computational biology. And for that there will be a faculty who is going to deliver nicely. You will say Chitto will be there. I think he's there in one of the speakers. So he is going to speak. He is a bioinformatician, so he can speak in that language. I think you will be comfortable. So Shohini's work was to go for the target. First target was specified, and now go for the plant extracts. Now the plant extracts, what? You know, ashwagandha. So that is Withania somnifera, the scientific name. And there was another. She found out that this Withania somnifera has two compounds. The major she had to extract, she had to screen so many compounds. Out of that, she found out that these two are there, which is very promising. So, therefore, and she created a library. Okay, I want to just, some terms I need to tell you. That when it is, suppose this is a receptor or that target, and this is a drug that comes. So this drug is called a ligand. So this is a receptor and this ligand, this binding has to be established in the most scientific way with the minimum binding energy. So then only it will be favorable. And you have to simulate the entire thing. We call it MD simulation. You have to mimic the living system in the machine, whether the drug will be stable or not, whether the betaferrin A or betanolone can be stable or not for a longer period, just mimicking. So this simulation and all, I think you are very, very conversant with, which I find that computational biology is now, we do a lot of simulation. So all of these binding and chemical identification, finally we were able to find out, yes, EIF43 is possibly one of the targets, if not the only target. So we have to find out that we have to do some wet lab. Wet lab means what? You have to do it in the cell system and see the gene expression which is going up, is going down or not, or if there are any other genes which went up, they are going down or not after the treatment. So these are the research that has to be done in the lab just to prove that the drug is druggable or not. Means, drug-like properties have to be determined through the software, through these servers, where a lot of data is stored and AI comes into this field. So finally, this part is there in clinical implications in glioblastoma for betaferrin A, betanolone, which is that ashwagandha. So that is, we have been able to prove it. So we had a paper also in that. I think the paper was shown over here. Wait, just a sec. Okay, this is missed out. JBSD. This is a good paper. So she had now, and the last part of my talk, just 10-15 minutes I will take your time. Please you just bear with me. Now what you see, evaluation of Vithania somnifera crude extract on EIF43 expression and functional toxicity assessment using Cinorhabditis elegans model. Very long title. And what is this? But if you forget about that Cinorhabditis elegans model, the rest of the words are very simple. We are taking plant extract like from Vithania somnifera, that plant we said, the ashwagandha, and EIF43 as a target, whether we can really use that drug or that medicine or that plant extract in treating cancer cells or in the cancer lines, can we treat directly? The question is yes and no. Why yes? Because if you treat the cancer cells, the cells might die. But you have to again do some doses and reduce the dose, and we say dose curves, and then finally you end up with one dose and you do all your experiments on that dose. So we got LC50 or LD50, whatever you call it. But that is a cell, a monolayer cell. It is not an animal. Now you cannot test that in human. Ethical issues. Nobody will say, no, I will not sign. I will not take that plant if I die or something problem happens. So using that same plant extract into human is just next to impossible. Next, who is the guinea pig? Rat, mouse. That also there is a lot of regulatory embargo. You cannot issue rat, mouse. Now where will I test? Who is going to be the guinea pig for that? So science, we have our answers. So there is some answers for such questions. I don't want to go to the scientific part of this target. It is a protein, very important protein, which really controls our translation, transcription. I think many of you know that gene expression, transcription, see replication, transcription and translation. So this central dogma, if everybody remembers their biology, so you remember? So this protein has its important role in this process of translation, transcription and translation. So if there is any problem with that, obviously it will impact the entire cellular mechanism. This is the paper. Okay, there I was trying to tell you that where is the paper. So this is the paper which, yes?
I just have a question.
Yes.
This plant extract is applied in a good form or pure form?
See, the one which we have already So I think in research, all the studies have to be done, the extract also and also the pure form, means the crude and the pure form, which pure form will give you the scientific data in a very precise manner, but the crude form may not give it. Because, absolutely, you're right. So you should keep your objectives open to all, because it is plant extract, so you have to be depending on the natural sources. Okay?
And another question. Why the C elegans is
Yeah,
selected
just-
higher than the zebrafish?
Okay. Just now, because I'm going to advocate on C. elegans now. So that's a very good question. Why C. elegans and not zebrafish? So he knows. If others do not know what is zebrafish is another model for research in the laboratory. But between C. elegans and zebrafish, the difference is one is an invertebrate and one is a vertebrate. You know what is a vertebrate and invertebrate. We have the vertebral column. So zebrafish is more mimicking like the human, because they have the vertebrate. But this C. elegans is a nematode, kecho. Okay, but it is microscopic. You know very well that there are many nematodes which are very big. The ones which infest our system, they are very big. They are macroscopic. But this worm is very fancy, very fancy. But I just want to get into it. So this was something that I want to let you know, that this is a worm. We call it a worm. C. elegans. The scientific name is Ceratodactylus elegans. It's very elegant to look like a snake, serpentine, and it feeds on bacteria and is cultured on the plate. If those who knows a little bit of microbiology, they know that on the plate you have to put some bacteria, they will grow, and you just release the worms into it, and everything is under microscope. You cannot see it through naked eyes. That is a problem. And it is an invertebrate. Now you might say, why am I working with an invertebrate to answer questions of human being? This is the most important question it should strike you. That I am going to treat human and I am working with a nematode. How does it help? Question is, the answers are over here. First of all, this is a nematode which has the gene, the sequence, homology almost more than 60% same as the human. So we call it homologous or homology. So if you use those terms in our jargon or in our term, it is a homology that they share a lot. So if you work with the worms, you can get some answers of the human. That is the answer which I will go deep into it. And the zebrafish that you say, now you might say, ma'am, why not zebrafish? Of course you're most welcome to do a zebrafish. I don't work with zebrafish, so I don't want to talk about zebrafish, but zebrafish is definitely a very, very good model. See, who is a good model? Now you will say, who is a good model? Somebody who looks beautiful? No! The genome is known to you. The sequence of that organism is known to you. It's in the red. It is in the public domain. So if you know the sequence, you know most of the diseases, the disease-causing genes. That is the beauty of all the model organisms, which includes not only Homo sapiens like us, not only rattus for the rat, mus musculus mouse, Drosophila fruit fly, the Drosophila. So those Drosophila, that is also another organism, and nematode is one. The C. elegans is a beautiful model, and why it is so popular these days, because this was the first multicellular organism, metazoa, whose genome sequence was published. So that is why C. elegans became so famous. But I am not advocating for C. elegans only because of genome sequencing, because I worked with it and I am very comfortable with it. Now the question is, why this title was there? Because this was a talk of one title when I was asked to give a lecture. 4 Nobels were awarded to C. elegans research, 4 different Nobel Prizes, and the model was C. elegans. And the fundamental research that was carried out, for that the jobs, I was kind of like pushed away because they were thinking, C. elegans is a fancy model. You should work with yeast. You should work with bacteria. And I said, this poor nematode eats bacteria. You see on the plate how it's eating the bacteria? It eats bacteria, but I'm comfortable with it. So anyway, I could not impress the experts over there. So I had no, and I have no complaints now. Now, the more facts about it is that it's a very simple organism, an animal, but you have to see it under the microscope. Looks like a snake. It is transparent, so you can see everything into it. And it has just 902 cells. Think of any other animal or any organism. This precision is not there. Just 902 was 900. 302, 952 I think, I forgot. 902, huh? So 302 is all taken away by the neuronal cell. So you know the cell lineage is very specific. But that is also not so important. Later on what happened was, see how it grows? These are the eggs and that is the larva, and that's how we call it embryogenesis. So the whoever knows biology will know what is embryogenesis. I think everybody knows embryogenesis. So that's how even our fetus, everything, they grow in our mother's womb also in this way. So that's how the worm grows. But the most important is in 2002, the first Nobel was awarded working with the C. elegans for developmental studies. Fundamental word, if anybody knows biology, they know apoptosis. Programmed cell death, that was discovered in working with the C. elegans. So all these scientists, Sidney Brenner, Bob Horvitz and John Sulston, they are just, Sidney Brenner is no more, but they were the pioneers. So 2002, I already returned to India, and I was very happy. At least now if I go for an interview, people might say, oh, C. elegans, because apoptosis, then the story doesn't end. This was another landmark, and I'm very happy to say this, you have heard about gene editing. I do not know whether you know anything about gene silencing, the word gene silencing. So gene silencing, if you do not know, it's okay, no problem. The gene is there to perform. If you silence it, then it will not perform and there will be some problem in your system. So what was the model to silence gene and see what is the effect and how the silencing was happening? They found out the mechanism. So Andrew Fire and Craig Mello, I would rather say I am very happy that Andy, we call him Andy because my postdoc boss did his postdoc under Andy. So we are kind of connected with Andy for a long time. So that was one thing. And I want to share, he is a mathematician turned biologist. No wonder Nobel comes not only from one single mindset, it comes from a lot of experience and expertise. So here, and another Nobel, of course, this is a little bit of fundamental, and that is Victor Ambrose and Gary Ruffo. Again, some molecules discovered in the worms has made a landmark story, microRNA. We say microRNA, small RNA. They have a lot to do in the system. So, why I am saying C. elegans? Because this is another good model to work and see whether you can have some ideas with whatever topic you have, and if you want to translate them into the living system, whether worm can be a good platform or not. And you see this? This is called green fluorescent protein, GFP. From where it is purified? From the jellyfish. Jellyfish is found in the sea, and they will grow. What the scientists did? They took the gene from the jellyfish, expressed it in another system, and if you just insert that gene into any organism, it will start glowing green. But the mechanism of this GFP was first discovered again in the C. elegans. So you see, this is I took some pictures when I was doing this. This is data from my paper.
The previous slide was showing something happens in one organism.
Yes.
What is the transformation?
No, this is what you know. All these dots that you see, these are neuronal cells. Okay. GFP is expressing in all. Over there you see the neural cord, GFP is expressing. And this is just to show you the movie was taken that the worm is moving and how the connectome is like kind of being displayed under the fluorescent microscope. It's just to depict like what are those cells which are expressing GFP, and whoever has taken, this is not my picture, this is my picture, these are from my paper where I've shown that the gene I was working with is expressed in the neurons, it is expressed in the muscles. So the muscle is glowing green and the neurons, do you see this is called the neuronal cord. So you will drive the expression of the GFP in the neurons. You are driving the expression of the GFP in the different regions of the animal. But it is not the GFP that you are going to express. It is your gene which is going to be expressed. GFP is just a reporter. I am the gene, I am the protein, I am carrying the GFP along with me. So wherever I am moving as a tag, this green is being transferred. So GFP is known as a reporter gene. Whoever is doing cell biology, they would know. So my purpose is to just let you know that there were 4 different researchers with the worms that has accorded the Nobel, and this person, this Martin Shafi, these are the 2 Japanese scientists who have, those are the chemists, and Martin Shafi was the biologist who conducted this work entirely in C. elegans to show that GFP can be used as an important thing, and it became now it is used in all cell biology. So they got the Nobel. So these four beautiful Nobels given according to the worms had made at least people like me who works with the worm has something to offer and can convince the audience or the students that you can work with the same. So now coming back to my original topic, Shohini was working with the glioblastoma. Now, if those bithanolide or bitaferrin A, those plant extracts, whether it can be used in human, first you should work with the animal model. Now, as you said that why can't we use it in the zebrafish? It is a vertebrate, it is a fish, it will be raised in a tank, in water, whatever. And this particular organism is cultured on plate. They eat bacteria, and you just have to see it under the microscope. The question is, treating in a monolayer cell like cancer cell, which has no tissues around it, and treating something in an organism which has the entire system, has different meaning. So if you are treating, if I am taking a drug, it is working in a system. If I am treating the drug with an animal like rat or mouse, this is working in a system. But if I am taking a tissue out of it and culturing the liver cells or the cardiac cells and I am culturing it, they are not animal full life, they are just monolayers. So getting the data out of it can give you the first-hand information, but not be a complete information. So animal model is always preferred. So now in my case, I am working with an invertebrate model. Many people are working with vertebrate model. So everybody has their own models because it's easy to culture. Only thing is that the requirements are slightly expensive because you need microscopes, you need incubators, you need many other gadgets which are slightly fancy other than the zebrafish and all. Okay. So that's how the worm would look like under the microscope. You see how they are serpentine, and if they are dead, they look like this. Now, if I am treating the worms, so same way, EIF43. Now let me get back to the original stuff. EIF43, a target which I have assigned to Shohini from the glioblastoma. I wanted to see whether worms also have EIF43. Yes, they have. They have EIF43, the same gene they have, but does that mean that I'm going to create glioblastoma in my worm? No. It has the gene, it has the protein, might be it has some different function. In the humans, it is a normal protein which has a lot of roles in transcription and transmission. Same way also in the worms, but only thing, worm is serpentine. It has some other way of living. Its biology is different than what we have. So you can always see that's how it grows. This is and that's how it looks. These are the eggs, and she is a hermaphrodite. She is a hermaphrodite, means she has both the egg laying and the sperm generating capacity. Both she has. So I'm to see how, so we are testing now that question, somebody has a root extract, right, a pure compound. So these are all with the root extract. See how the worms, when they die, they look like this, and when they are living, they look serpentine. So it's just to give you an idea that, see, we are integrating biology, we are integrating living system in your research. See if we can make a marriage out of this two and make it a successful one. So that's the purpose of showing you. It's not that I'm giving a talk on EIA 483, I'm just trying to make you understand how we predict a target and how we go about it. And pharmaceutical industries will always look for targets. And our FDP is mostly focused on this aspect, that it is a pharmacy precision, okay, how AI is integrated into it, that is our part. So I just wanted to share that we do have AI in our research domain. A part of it is, it's not all AI, but definitely a part of it is definitely driven by AI. So now those experts in AI, what you can do is, this is my kind of suggestion, that how you can make AI type like computer vision, deep neural networks, transformer models, spiking models, all of these models that you see can be applied into the C. elegans research in various ways. And it has been carried out. Trust me, I'm just started a little bit. The tip of the iceberg. I will not even say that I have done a lot. Little bit, that also is not so significant. I have to work a lot, my students have to work a lot to be able to do something worth it. But at least for the new generation who are aspirants in formulating some new ideas, applying AI in the living system, at least C. elegans can be one. You can try out. So these are some of the applications, because the worm moves, it has all the biological parameters, so why not try it? So this is just to give you an idea how you can integrate AI in C. elegans research. This is just to give you an idea. And finally, AI is in pharmacy, whether it is boon or bane. I don't know, I leave that question to you. You might be thinking that we are going to lose our jobs. No, never. Definitely not, but it has really answered many questions in its own way. And we have to think that, yes, it is a boon. At least I will advocate for it as a boon. But if you have any other, what do you call it, feedback, you're always welcome. I am always very much appreciative about the knowledge that AI has really transferred into the different domains of science and other disciplines, and has made our life much practicable in a way. We are not going to lag behind. Now the question is, if you think that no, it is not really, we should not take AI, we should discard AI, then yes, okay, you do it, you're most welcome. Do it, and the next country will go ahead with it. You will just see. You will purchase all items from the foreign country, all manufactured in yours, because they are benefiting out of our produce. So don't lag behind. I will say, be positive, be very you should always consider all the ethical issues. I'm not saying that everything is correct. Keeping in mind that all the regulatory bodies, all we need to have is that you have to abide by certain principles, certain rules, and I think it is applicable across all the domains. All rules and principles only should not be restricted to AI. It is in every discipline that we follow. Every discipline has some ethical issues, and we all have to abide by it. Nothing doing on that. So that's it. Now is that no coffee, no problem. I just wanted to my slide has some coffee to kind of drive you. So I'm already at the end, and you have another session, and I'm happy that I could make it on time. One thing I want you to all do sorry that I've taken away a lot of 2 hours. Maybe you could have studied something different or known something different. You could have, maybe it was not interesting for some, maybe it was interesting for few. So I don't know, I did my job. Whether I did justice to this session or not, I am really not aware of it till I get a feedback. But one thing I want you to do is, I have just asked, I am throwing this, some group activity. I'm leaving this slide, I can share it with you. And I will be happy that after 2-3 days, I am leaving I want you to just, I have my WhatsApp number and my email ID. And the group ID, the group activity that I'm kind of expecting that you have, you divide into your groups. I'm leaving up to you. And I want you to make it interdisciplinary because you are all from engineering background. 1 and 2, how many healthcares are there? 1, 2, 3, 4, 6, 7, 8. So there should be 9 groups. 9 groups, at least 1 healthcare should be there with all the engineers. Okay, and devise some simple projects. I don't have to give me a project, just the bullets will be doing enough. You try and devise some projects related to healthcare, related to AI is there, so do some studies by yourself. And just in bulleted 1 page, 1 A4 page, you just put your group name and your names and send it to WhatsApp it to me. I just want to see that whether you could gather has a pause. And for that, all who are responsible, genes, protein, gene expression, each one of them. So it is the duty of the researchers. See, doctors who are into very deep into research, they know. So if there are side effects, you need to jot down, first of all, what is the immune response, what are the players who are responsible for that effect, and then first you have to treat it general. If it works, fine. Otherwise, if you take each target, like every pathway, every protein has a role to play. So the overall response can be diarrhea, overall response can be hypersensitive or allergy. But an allergy, it cannot be from one particular cause. There are many roots to cause that allergy. So it is again the duty of the researchers to find out. So if there is some AI system and they jot down like homeopathic doctors do, they will ask you such questions that you feel embarrassed. I have seen doctors asking such questions, but I now understand that they have a meaning to it. And it is true that many doctors, they don't consider, they just prescribe something and it doesn't work, and then again we go back to the same doctor. So the answer is definitely general in the beginning, but later on it will become personalized. So treatment, if now personalized medicine is now, the research is going on, maybe after a few years it will be in the market. You will have your own medicines. Means if your genome sequence is known, if you dump your sequence into a machine, the machine will automatically do some calculation and tell you that you are prone to this disease. What a beautiful way to analyze, and can't we wait for those years? I think all the computer professionals, please come up and with some ideas and make us smarter. Because those machines will be smart, but you have to govern it. Mind you, do not allow the intelligence to capture you. Your brain is there, you have the personal touch, but definitely it is going to help, but not going to remove your presence or your importance ever. Human presence and human participation is definitely required. It's only our life is going to be little bit simple. Okay, so I've done my job. Yes. Yes.
Madam, any other
question? Yes. Please, whether if you have, I know if you have questions, you're most welcome to call me up or mail me. That would be easier. Please.
Ma'am, you have shown that C. elegans has got antibacterial activity. So,
antibacterial, no, it eats bacteria. I won't say it survives on bacteria. But yes, what is your question?
Can C. elegans be used instead of antibiotics, or it has not come?
No. on bacteria. What you can do is, there are many pathogenic bacteria. Whoever is working knows bacteria, they will be able to know. Then you have pseudomonas, you have cholera, Vibrio cholerae. Every bacteria, those which are pathogenic, which causes diseases, you need to know what are the agents or what are the players, how it is affecting, how the diarrhea is getting, is being caused in the human. We know that if cholera bacteria enters, we will have diarrhea. And the protein has also been identified whose expression actually gets distorted because of the toxin that is there. Worms, C. elegans can be a platform to test all the pathogenic bacteria and see what is happening to the worms. The worms will either die or will not lay eggs, will have a lot of physical and behavioral anomalies by eating that pathogenic bacteria. It is not antibacterial. Mind you, it is living on bacteria, and the bacteria is simple E. coli. With the strain name is OP50. E. coli, very non-pathogenic bacteria, and this worm is growing at 20 degrees. So you see, I need an incubator, a small fridge type, which is maintained at 20 degrees continuously. My worms are very, very fancy in a way, very delicate. And it was once upon a time a soil organism, now it is a lab organism. And I won't say it is very expensive, but the instruments and all are slightly like unique. But it is not antibacterial. Yes, very good question. I appreciate that. That means that it eats bacteria, you can test many bacteria on worms.
We can see the toxic effects of the bacteria.
Absolutely, and there are beautiful papers, But my boss, my Korean boss, when I was coming back to India, I said, what am I going to work? So I said, go back. 2001 was the year. He told me, see, I understand India, there are many limitations in funds and all, because over there the fund was not a big problem. So he said, why don't you start with pathogenic clinical, all those samples which are found in the hospitals? You simply get those bacteria, but for that you need a biosafety level lab set up, which is not possible in any university. You can, but you have to have a separate unit. Cholera, NICET, cholera, all these institutions and all these hospitals, and you have a special unit. In a public university, very difficult to handle these medical samples. Very, very difficult. So again, ethics. You need human ethics, you need animal ethics, all the ethics. Yes, it is there. But I said that my Korean boss, I said, I'll try, but I have to collaborate with hospitals. And coming to a country back, to my homeland, and then starting a new project was definitely challenging in that. So talking about 25 years ahead, back. So just for my just to let you know that I was now I want to just be very free. I had been in Makaut from the day one, almost like I'm the first faculty, with the biotech department opening up with my entry, and that was the first academic department. So I joined in 2002, okay, and it was in October 4th. I'm going to complete my 23 years in this October, and yes, I was in IICB a year before that, and when I came back from Korea, I had lots of plans, but yes, thanks to my husband, he is also associated with a similar type of, but yes, he's a scientist. He's now retired, a scientist in IICB. So he, Arun Bandyopadhyay, he was a director. He retired as a director. So he told me that I can offer you my lab setup for the cell culture. So I had to start with my cell culture. I could not start with C. elegans, And then I went back for 1 year again sabbatical, and then I started my research in C. elegans in Makaut, WBUT that time. And thanks to all my students who really had really tolerated me, because I was in the administration for so much time, had to devote, that my research got lagged and lagged and lagged. and I am going to complete my 60 years this November. I'm going to enter into the senior citizen very soon. But then, yes, I don't think that we should stop. And if you young minds out there, if you can bring some ideas, you are most welcome to collaborate with us, and I will always try to help you out. Okay? So with this, once again, thank you very much. Coffee! Hey, coffee দিয়েছে। Coffee না আগে দিলে না আমার প্রজেক্টটা বেরিয়ে যেত। না না, joking. এই গরম আছে হ্যাঁ, কি বলছো হ্যাঁ। আচ্ছা, now we can stop in English. Everybody understands Bengali? But I think recording should stop now.